The quest to unlock the secrets of human longevity has transitioned from the realm of speculative alchemy to the precise corridors of molecular biology, centering on a coenzyme so fundamental to life that its depletion is now considered a primary hallmark of aging. Nicotinamide adenine dinucleotide, or NAD+, functions as the essential cellular currency, an indispensable electron carrier that facilitates the conversion of nutrients into functional energy while simultaneously serving as a critical signaling molecule for DNA repair and genomic stability. As we traverse the decades of our lives, the systemic levels of this vital molecule undergo a precipitous decline, often plummeting by more than 50% by the time we reach our sixth decade, a phenomenon that triggers a cascade of cellular dysfunction ranging from mitochondrial decay to the exhaustion of stem cell populations. This bioenergetic crisis is not merely a byproduct of time but a fundamental driver of the physiological erosion we identify as aging, leading researchers to explore how we can biochemically intervene to restore youthful NAD+ concentrations. While the pharmaceutical industry has rushed to develop synthetic precursors and expensive supplements like NMN and NR, a groundbreaking shift in nutritional science is revealing that certain complex food matrices contain potent bioactive compounds capable of stimulating endogenous NAD+ production more effectively than isolated pills. These natural interventions do not simply provide raw materials, they modulate the very enzymes responsible for the synthesis and salvage of NAD+, effectively retuning the cellular machinery to maintain a state of metabolic resilience. To understand why certain foods can outperform synthetic drugs, one must first appreciate the intricate dance of the NAD+ salvage pathway, where the cell recycles nicotinamide back into active coenzymes, a process that becomes increasingly sluggish with age. The degradation of NAD+ is largely driven by NAD+ consumers, like CD38, an enzyme that increases with chronic inflammation and literally consumes the cell's energy reserves, leaving little for the sirtuins, the so-called longevity genes, to perform their essential tasks of silencing pro-aging genes and repairing double-strand DNA breaks. When we introduce specific, high-potency phytonutrients through a targeted diet, we are not just eating, we are engaging in a form of biological programming that inhibits these wasteful consumers while upregulating the rate-limiting enzymes of the salvage pathway. This internal restoration provides a more sustainable and holistic elevation of NAD+ levels compared to the spike and crash profile often associated with high-dose synthetic supplementation, which can sometimes lead to an imbalance in methylation or the accumulation of metabolic byproducts. Furthermore, the synergy found in whole foods, where antioxidants, fibers, and polyphenol work in concert, ensures that the increased cellular activity resulting from higher NAD+ levels is supported by a robust defense system, preventing the oxidative stress that can sometimes accompany a sudden surge in mitochondrial metabolism. As we delve into the specific foods that harbor these life-extending properties, it becomes clear that the future of anti-aging medicine lies not in a laboratory-synthesized capsule, but in the sophisticated biochemistry of nature, harnessed through a deep understanding of how specific molecules interact with our genetic architecture to turn back the clock at a cellular level. The transition from a state of NAD+ deficiency to one of abundance represents a fundamental shift in the aging trajectory, moving us away from a future of frailty and toward a paradigm of healthspan extension, where the vigor of youth is maintained long into the later years of life through the strategic application of nutritional science. This foundational understanding sets the stage for identifying the three specific nutritional powerhouses that stand as the most effective biological keys to unlocking the body's latent potential for self-repair and energetic renewal, transcending the limitations of modern pharmacology. The strategic elevation of NAD+ levels within the aging human body requires a dual-action approach that moves beyond simple precursor supplementation, focusing instead on the preservation of existing cellular pools and the optimization of recycling mechanisms. Central to this biological strategy is the neutralization of the enzyme CD38, a pervasive glycoprotein that functions as the primary consumer of NAD+ in mammalian tissues. As the body ages, a state of low-grade, chronic inflammation known as inflammaging triggers a dramatic upregulation of CD38 expression, particularly within the immune cells and the vascular endothelium. This enzymatic surge acts as a metabolic sink, aggressively breaking down NAD+ before it can be utilized by sirtuins for DNA repair or by the mitochondria for ATP production. Research in geroscience has identified a specific class of polyphenolic compounds, most notably apigenin and luteolin, which are found in high concentrations in specific vegetables like parsley, celery, and radicchio, as potent naturally occurring inhibitors of CD38. Unlike synthetic pharmaceuticals that may have systemic side effects, these bioactive flavonoids bind to the catalytic site of the CD38 enzyme, effectively slowing the rate of NAD+ degradation and allowing the cell to maintain a youthful energy budget. By integrating these specific botanical agents into the daily diet, an elderly individual can fundamentally alter their internal biochemistry, shifting the balance away from catabolic decline and toward homeostatic maintenance. This process is not merely about providing a building block but about repairing a leaky bucket in the cellular reservoir. When CD38 activity is suppressed, the availability of NAD+ for the SIRT1 and SIRT3 enzymes increases exponentially, leading to improved mitochondrial biogenesis and the activation of autophagy, the cellular cleaning process that removes damaged proteins and organelles. Furthermore, the second pillar of this nutritional triad involves the activation of the nicotinamide phosphorybosyltransferase transferase (NAMPT) enzyme, which is the rate-limiting step in the NAD+ salvage pathway. This pathway is the body's primary method of recycling nicotinamide, a byproduct of NAD+ consumption, back into a functional coenzyme. Scientific investigation has highlighted the unique properties of fermented sprouts and specific germinated seeds, which contain high levels of bioactive molecules that stimulate NAMPT expression. This stimulation ensures that the recycling loop remains robust, preventing the accumulation of nicotinamide which can, in high doses, actually inhibit sirtuin activity. The synergy between inhibiting the wasteful CD38 enzyme and accelerating the efficient NAMPT recycling loop creates a metabolic environment that synthetic pills often fail to replicate. This holistic approach addresses the systemic nature of aging, where various pathways are interconnected. For instance, the reduction in CD38 activity not only saves NAD+, but also reduces the production of cyclic AD-pyribose, a molecule involved in calcium signaling that, when imbalanced, contributes to cardiac and cognitive decline. By focusing on these specific food matrices, we are leveraging millions of years of botanical evolution to communicate with our genome, sending signals that promote survival and repair over decay and senescence. This dietary intervention provides a steady, sustained release of bioactives that align with the body's circadian rhythms, unlike the bolus doses of supplements that can disrupt delicate metabolic equilibriums. The efficacy of these foods lies in their complexity, providing secondary metabolites that support liver function and gut health, ensuring that the liberated NAD+ can be effectively transported and utilized by the most energy-demanding organs, such as the heart and the brain. As we transition from the theoretical understanding of these mechanisms to their practical application, it becomes evident that the first two superfoods in our triad, high apigenin botanicals and NAMPT-stimulating fermented germinal matrices, form the foundational defense against the biological erosion of time. They represent a sophisticated form of nutritional pharmacology that empowers the aging organism to reclaim its metabolic sovereignty, setting the stage for the third and most powerful food component which bridges the gap between raw energy production and the ultimate protection of the genetic code. The final and perhaps most revolutionary component of this nutritional triad involves a shift in focus from the production of NAD+ to the absolute protection and optimization of the organelles that utilize it, the mitochondria. While the first two phases of our dietary strategy focused on slowing the degradation of NAD+ and enhancing its recycling, the third pillar centers on the introduction of longevity vitamins that are rarely found in the standard modern diet but are abundant in specific fungi and ancient grains. Chief among these is ergothioneine, a unique sulfur-containing amino acid derived primarily from mushrooms, which humans possess a highly specific transport protein for, known as OCTN1. This evolutionary trait suggests that ergothioneine is not merely an optional nutrient but a fundamental requirement for cellular stability. In the context of the aging cell, the increased presence of NAD+ can significantly accelerate mitochondrial respiration. However, without a potent intramitochondrial antioxidant like ergothioneine to quench the resulting reactive oxygen species, this heightened metabolic activity can lead to oxidative burnout. By consuming high concentrations of ergothioneine-rich mushrooms such as king oyster, shiitake, or porcini, an elderly individual provides their cells with a biological shield that migrates directly into the mitochondrial matrix to protect DNA and membranes from the very energy production process they are trying to enhance. Parallel to this is the role of spermidine, a polyamine found in abundance in wheat germ and aged cheeses, which acts as the ultimate master switch for autophagy. Autophagy is the body's innate cellular garbage disposal system, a process that declines with age, leading to the accumulation of zombie or senescent cells that secrete inflammatory cytokines. Spermidine mimics the effects of caloric restriction by inhibiting acetyltransferases, thereby triggering a deep cellular cleanse that removes the protein aggregates associated with neurodegenerative diseases and cardiovascular stiffness. When these three nutritional forces, CD38 inhibitors, NAMPT activators, and autophagy inducers, are combined, they create a synergistic effect that no single drug can replicate. This is because aging is a multifactorial failure of systems, and the solution must be equally multidimensional. The integration of these foods into the daily life of an aging adult represents a shift from reactive medicine to proactive biological management. This holistic approach ensures that as NAD+ levels rise, the cellular infrastructure is clean enough to use that energy and protected enough to sustain it without collateral damage. The era of passive aging is over. We now possess the biochemical keys to unlock a future of enduring health, proving that through the marriage of ancient nutrition and modern science, we can indeed grow older while staying fundamentally young at the cellular level.